All right, 8.3 is going to specifically talk about the prep step. The prep step is the step between glycolysis and the Krebs cycle. In some books and in some just like websites or content, you might actually see this incorporated with the Krebs cycle, but we're going to specifically take this apart and look at it individually. We're also going to, in this section, going to look at mitochondria a little bit more deeper. So let's take a look here. Mitochondria, remember we have that double membrane. That outer membrane is the more eukaryotic, the inner membrane is the more prokaryotic of the two. That's because a prokaryotic cell billions of years ago was enveloped by a eukaryotic cell, and that was called the endosymbiotic theory. Matrix, this is the liquid inside here. You can see where the matrix is labeled. This is the liquid inside of the mitochondria in the inner membrane. This is like the cytoplasm of the mitochondria. The cristae, these are folds within the inner membrane. These increase surface area. If you think about how increasing surface area is good for a cell, you're increasing the amount of enzyme or reaction location in the mitochondria. This makes it a lot more efficient at breaking down just like glucose and other things. When you increase the efficiency, that's going to be a huge evolutionary advantage because you're getting more energy and quicker at that. Our inner membrane space here, you can see between the two membranes, that's an inner membrane space. We're going to talk about that and why it's important. It's highly important in the electron transport chain. But I would know more in depth about each one of these locations. Yeah. All right. So I have this awesome video in the lecture, and I didn't want to skip it, but it doesn't really play the video I'm sorry, it doesn't play the audio. So I'm just going to explain what goes on here. So this is the mitochondria you can see here. You can see all those ATP molecules coming out. Those are what's in red. What's really important here is the structure. We can see again, here is the or we have the outer membrane. You can see here, then you go to the inner membrane. Within the inner membrane is that mitochondrial matrix, as it's highlighted right there. That's that liquid inside of the mitochondria. The inner membrane space, that's the location between the two membranes. You can also see these cristae that are just those folds on the inner membrane. Now it's going to show you again what what each location looks like a little bit more in depth. We have two membranes. We have the inner membrane space, we have the cytoplasm, and we have the matrix. This is where a lot of the ATP is going to be produced on that inner membrane. Prep step is also known as pyruvate decarboxylation, and I know they're going to give a couple names for each step. It's just what it is. But let's talk about why it's called pyruvate decarboxylation. Pyruvate. We're going to start out with pyruvate. You can see here this pyruvate molecule. Now, with this pyruvate molecule, understand that I am not looking for you to understand this whole structure. I just want you to know that there's 3 carbons here. Decarboxylation. If you carboxylate something, or you go through carboxylation, you're going to add a carbon. If you decarboxylate something or go through decarboxylation, you're going to take a carbon away. Here is a big hint. Please remember this. Anytime we lose carbon in cellular respiration, we're losing it in the form of CO2. And you can see here, this carbon with these oxygens are going to leave in the form of CO2. You're breaking down glucose, you're breaking down these organic molecules and you're producing CO2 that you breathe out. So for every pyruvate, you're going to lose 1 CO2. You're going to reduce 1 NAD and make an NADH. That's going to, before we get to there, again, we have this NADH. This is going to go somewhere else. But again, we have this pyruvate. We're going to lose this carbon dioxide. This acetyl group, so right here, this is called an acetyl group. This is going to bond with something called coenzyme A. Coenzyme A is going to be a molecule that helps acetyl-CoA go into the next cycle, which is the Krebs cycle. Now, technically, the pyruvate is in the cytoplasm. It has to cross 2 transport proteins to make its way into the mitochondrial matrix. There is where the reactions are going to occur. All right, so I love this meme because it's just so perfect. The acetyl group that we're talking about that's really specific, we have 2 carbons here. This is a small molecule. Coenzyme A is going to be, it's a coenzyme. It's just a helper with, it's going to help with in the Krebs cycle. But you can see this is a massive molecule here. It's just going to aid. I just love Patrick right here, just smiling his acetyl group. The acetyl group is just very small. The coenzyme A is very big. This is going to help acetyl go into the Krebs cycle. And again, we have our coenzyme A here and our acetyl group. just too perfect. Like I said before, we're going to have that pyruvate move through both membranes of the mitochondria through a transmembrane called pyruvate translocase, which is again just going to move in pyruvate into the mitochondrial matrix. This is where it's going to go through the reaction. We're going to get acetyl CoA out of this, which is going to go into the citric acid cycle, or it's also known as the Krebs cycle. All right. So again, I'm just going to review this. Pyruvate is going to go into the mitochondrial matrix. It's going to break down and break off a CO2 molecule. We're also going to get an NADH out of here. We're also going to attach a coenzyme A to the acetyl group that's left over, and that's going to go into the Krebs cycle. Now, this whole process has to occur twice because we're getting two pyruvates from glycolysis. So after we have one pyruvate go through this process, another one's going to go through this process. So essentially, we make two CO2s, two acetyl CoAs, and two NADHs.